16.8.2 Apiary management. Seasonal management of honeybee boxes. Seasonal management is most important and essential part of apiculture for the better production of honey, pollen, Mishra, 1995. The abundance of bee flora and their continuous availability is one of the major prerequisites for successful beekeeping. This enables the beekeepers to exploit these sources to the maximum utilization by the bees. Every region has its floral dearth periods of short or long duration, and it is essential to manage the honeybees and beehives in the dearth period, as during this period, there is lack of availability of nectar and pollen in flora required for quantitative and qualitative production of honey. Acratinocle, 1987. To overcome this problem, it is important to find out the suitable bee flora available in the locality to propagate and manage the plant species with the option of abundant nectar and or pollen to overcome the problem during the dearth period. For commercial apiculture, pollens and nectar are available only during certain period. When surplus food source is available is known as honey flow season. In contrast, during dearth period there will be scarcity of food. Suitable season for starting beekeeping coincides with mild climatic conditions and availability of bee flora in plenty. Normally, spring, February, April, and post-monsoon, Sep. November seasons are the best periods to start beekeeping. Indian seasons are spring season Vasanthratu, mid-February, mid-April summer season Grishmaratu, mid-April to mid-June monsoon season Varsharatu, mid-June to mid-August autumn season Sharadratu, mid-August mid-October pre-winter season Hemantratu, mid-October mid-December and winter season Shisharitu, mid-December mid-404. February. Various operations required to be undertaken for augmenting colonies' productivity are given below. Spring management. Management operations to be undertaken during spring are given below. 1. Examination of colonies. On some warm and sunny day, examine the colonies quickly and carefully with least exposure to the chilling weather and robber bees. 2. Equalizing the colonies. The colonies can be equalized by A. Substituting the combs with food reserves, supplementary feeding. B. Providing the emerging bee combs. C. Uniting the bee combs colonies. D. Giving young bees to the weaker colonies. 3. Provision of space. During spring, the colonies enhance the brood rearing. Hence, there should be no dearth of space to cope up with increased egg laying by the queen bee. 4. Swarm prevention and control. A. Examine the colonies and remove congestion. Provide more drawn combs, comb foundations, supers, etc. 5. Control of mites and brood diseases. Examine the symptoms of various mites and brood diseases. On spotting any, take appropriate management measures to contain the menace. 6. Colony multiplication and commercial queen rearing. March-April is the best season for colony multiplication and commercial queen rearing. Improve the existing stock by selective breeding of best-performing colonies. Mass-reared queen bee can also be used for multiplication of existing stock and also for replacement of older queen bees, requeening. 7. Extraction of spring season honey. Multiple extractions of honey during this period are possible. Only ripe honey from 405. Broodless combs from super chamber should be extracted. 2. Summer management. Mid-April to June months are extremely hot. However, this is the major honey flow period too. The following operations need to be considered during the season. 1. Shifting the colonies to thick shade. Colonies should be moved to shady places every day by less than 3 feet. 2. Regulating the microclimate of the colonies. By using wet gunny bags over the colonies and sprinkling water around the colonies in the apiary during noon hours, temperature in the apiary can be reduced and humidity increased in hot and dry months of May and June. 3. Provision of ventilation. Improve the ventilation of the colonies to cope up with the respiration of the bees and hastening the honey ripening. 4. Provision of fresh water. 5. Honey extraction. Summer season honey can be extracted. 3. Monsoon management. Manage the colonies during this season as below. A. Ensure colonies placement on upland area and away from village water ponds. B. Clean and bury deep the debris lying on the bottom board. C. Keep the surrounding of the colonies clean by cutting the unwanted vegetation which may hamper circulation of air. D. Provide sugar feeding, sugar, water equals L if required. E. Check robbing within the apiary. F. Unite weak, laying worker colonies. Control wax moth, ants, wasps and bee-eating birds. IV. Autumn management. Important operations to be undertaken during this season are 406. A. Provision of space. B. Strengthening the colonies to stimulate drone brood rearing, if queen bee rearing is to be undertaken. C. Control of ectoparasitic mites, brood diseases, wax moths and wasps. D. Autumn honey extraction before the winter sets in. V. Winter management. Normally winter extends from December to mid-February, but this period may vary from region to region. During winter, very low temperature, westerly chilly winds, foggy cloudy days and winter hamper the bee activity. Brassica comes in bloom during January. To perpetuate the colonies through winter, following operations are generally required. 1. Colony examination. Examine the colonies on a warm, sunny day for the presence of queen, brood and food reserves. Open the colony for minimum time to avoid chilling of brood. Weak colonies should be united with stronger ones so that the strong unit overwinters well. 2. Feeding. If there is food scarcity or expected in the ensuing winter, feed concentrated sugar, water equals 1 sugar syrup, supplementary feeding, by filling in the drawn combs at the onset of severe winter. 3. Shifting colonies to sunny places. The colonies should be shifted to sunny places with hive entrances facing southeastwards. 4. Protection from the chilly winds. Plug cracks and crevices and narrow down the hive entrance. 5. Unite weak colonies with stronger ones. Follow newspaper method for uniting the colonies. 6. Removal of extra drawn combs and winter packing. Remove the extra empty combs and store them properly to save them from mice, rats. Depending upon the strength of the colonies and severity of winter, provide one or two-sided inner winter packing combined with need-based outer packing. 16.8.3 Controlling swarming. 407. Examine the colonies and remove congestion. Provide more drawn combs, comb foundations, supers, etc. B. Improve ventilation and provide required shade. C. Clip the wings of the laying queen. D. Use wire entrance guard, queen excluder at the bottom board. E. Reversing brood and honey chambers for mitigating congestion in brood chamber. F. Destruction of the queen cells raised due to swarming instinct. G. Dividing overcrowded colonies. 16.8.4 Handling of bees. Division and uniting of honeybee boxes. I. Division of honeybee boxes. Colony division is a method of multiplying bee colonies, i.e. producing 2 or more colonies from a mother colony. Colony division is used to control swarming as well as in commercial beekeeping to increase the number of colonies. Methods for colony division. I. Natural division using queen cells developed during swarming. The presence of multiple queen cells in a colony during the swarming season indicates a need for division. Dividing such colonies and using the queen cells in new daughter colonies can help control swarming. However, although it solves the immediate problem of swarming, it does not help improve the genetic traits. E. Colony division from queen production. Select the best colony based on the selection criteria given above. Produce queens from this colony before the onset of honey flow. These queens can be used to replace the old queen and to start new daughter colonies. The mother colony can be multiplied into several nucleus colonies, but each should have at least 2 brood combs and 3 to 4 combs with food, nectar and pollen. The prepared colonies can then be sold or migrated according to need. 408. B. Uniting of honeybee boxes. Uniting 2 colonies into one is done when one of them is weak or queenless or for other reason like bad traits, etc. Each colony has its own colony-specific odors and it is very difficult to combine the 2 colonies unless their odor is mixed well. Any attempt to unite these colonies without mixing their odor result in infighting and deaths will occur on large scale. Therefore, first step will involve bringing the 2 colonies into contact with each other. If uniting is done abruptly, the field workers of the colony shifted will not recognize the new place and returning to their original place will persist. This problem can be overcome by moving a hive gradually at the rate of 2 or 3 feet per day, so that the field bees get habituated to the changing position of their hive and will not drift back to the old site. When colonies are sufficiently close, 1 or 2 feet apart, they are ready for uniting. They can be united by 3 methods either 1. Direct uniting, 2. Newspaper method, 3. Smoking method. I. Direct uniting. The 2 hives to be united are brought near gradually and kept side by side. A queen with undesirable traits is removed from one of the hives. In the morning, when the bees are busy, the frames from 2 hives are gently inserted into one hive. Using this method successfully depends on how well it is executed. E. Newspaper method. Top cover is removed and the frames are covered with a piece of newspaper having a few holes made with a small nail. The bottom board of the upper colony is then removed and the brood chamber is placed above the other colony, the newspaper forming a partition between the 2. After a day or 2, the odors of the colonies will mix and the bees will cut through the paper and will unite together, forming a single colony. After a few days all the frames can be placed in one hive and the upper chamber can be removed. E. Smoking method. Colonies can be united using smoke method. When the colonies to be united have been 409. Brought close to each other, both should be smoked heavily and thin sugary syrup scented with oil of peppermint or wheat flour sprinkled over them. The combs with the bees of the colony to be united should be altered with the combs of the other colony. More smoke and syrup or flour should be applied and the colony closed. The work of the queen may be checked up after 3 or 4 days. It is better to unite a laying worker colony to several strong colonies by giving from 1 to 2 frames to each of them. If all its frames are united to one colony, there is danger of latter's queen being killed by the laying workers. 16.8.5 Extraction of honey and wax and other bee products. In addition to honey, beeswax, royal jelly, bee venom, propolis, and pollen are other important honeybee products. Honey is harvested after a flowering season. Beekeepers select combs with ripe honey covered with a fine layer of beeswax, usually those closest to the outside of the nest, in traditional or top bar hives. Honey extractors are used only to extract honey from super combs. Before placing combs in the extractor, the sealing of cells on the combs is removed with a sharp knife. Extractor should be worked slowly at the beginning and at about 150 revolutions per minute at the end for about 1 to 2 minutes. After that, the frames are reversed and the extractor is used again. A muslin cloth is used to filter the honey after it has been extracted. On the day prior to honey extraction, bees escape between the brood and super. Remove the escape as soon as possible after honey extraction. It is recommended to remove the super chambers during the lean season, May-September, arrange the healthy brood combs in the brood chamber, and use division boards to restrict the space. Feed artificially once a week with 1 sugar syrup in water. According to the size of the colony and the availability of stored food, each colony may require 500 to 750 grams of sugar per week. In the absence of natural pollen sources, pollen substitutes may be provided to the colony. 1. Honey. It is a solution of sugars and other minor constituents that bees collect from flowers and concentrate into honey. Honeybees produce this sweet, viscous fluid. Nectar is collected from 410. Nectarines at the base of flowers. Also collected from extrafloral nectaries, which secrete nectar other than flowers. It is collected also from fruit juice, cane juice, etc. Collection and ripening of honey. Depending on the nectar source, honey contains a wide range of sugars, as well as minerals, vitamins, proteins and amino acids. Nests near honey storage areas usually have a temperature of 35 degrees Celsius. A field bee draws nectar with its lapping tongue, known as a proboscis. The nectar collected by hive bees is regurgitated by field bees. There is 20 to 40% sucrose in nectar. Sucrose is converted into glucose and fructose by the enzyme invertase. Honeybees produce invertase in their saliva and in nectar. Finally, honey is ripened by enzyme action and evaporation of water due to temperature and ventilation produced by bees fanning their wings. When the water content in the cell reaches about 20%, the bees cap it with wax. Honey is now considered ripe and will not ferment. Bees prepare for themselves a concentrated food source in this way. Since the honey does not ferment, bacteria cannot grow in it, and it will not deteriorate over time. When there are no flowers, this food sustains the bees. 2. Beeswax. Beeswax is the material that bees use to build their nests. It is produced by young honeybees that secrete it as a liquid from special wax glands. Worker bees secrete a field bee draws nectar with its lapping tongue, known as a proboscis. The nectar collected by hive bees is regurgitated by field bees. There is 20 to 40% sucrose in nectar. Sucrose is converted into glucose and fructose by the enzyme invertase. Honeybees produce invertase in their saliva and in nectar. Finally, honey is ripened by enzyme action and evaporation of water due to temperature and ventilation produced by bees fanning their wings. When the water content in the cell reaches about 20%, the bees cap it with wax. Honey is now considered ripe and will not ferment. Bees prepare for themselves a concentrated food source in this way. Since the honey does not ferment, bacteria cannot grow in it, and it will not deteriorate over time. When there are no flowers, this food sustains the bees. 2. Beeswax. Beeswax is the material that bees use to build their nests. It is produced by young honeybees that secrete it as a liquid from special wax glands. Worker bees secrete wax when they are 14 to 18 days old. On contact with air, the wax hardens and forms scales, which appear as small flakes of wax on the underside of the bee. About 1 million wax scales make 1 kilogram of wax. Bees use the wax to build the well-known hexagonal cells that make up their comb, a very strong and efficient structure. Bees use the comb cells to store honey and pollen. The queen lays her eggs in them, and young bees develop in them. Beeswax is produced by all species of honeybees, although the waxes produced by different species have slightly different chemical and physical properties. Processing. Bee wax is obtained from the cappings collected during honey extraction. Wax is obtained from old combs that are unfit for use and from combs damaged during honey extraction. Best grade. 411. Wax is obtained from cappings where the recovery percent is higher. In India, major proportion of wax is from combs of Apis dorsata. 3. Bee venom. Sting of worker bee is attached to a poison sac where venom is stored. Newly emerged bee is unable to sting because she cannot insert the sting which is not fully chitinized. Also little amount of venom is stored in the venom sac. A bee, when 2 weeks old, has maximum venom in her poison sac. Production. Bee venom is commercially obtained by the use of electric shock. An electric current is passed through copper wires at 12 volts. The bees get shock, irritated and release venom by inserting the sting into a thin nylon cloth below the copper wires. Venom is deposited on a glass plate placed below the nylon sheet. The venom on drying is scrapped from the glass plate. 1 mellifera colony yields about 50 milligrams of venom. 4. Propolis. Propolis gathered by bees from resinous exudes of tree. In the bee colony, propolis is used for sticking frames, sealing cracks and crevices, but it is a contaminant of comb wax. Propolis is obtained by scrapping it from the frames. 5. Royal jelly. Royal jelly is secreted by gland of nurse bees of the age of 6 to 12 days when the glands are fully active. It is very nutritious food and is fed to the young worker larva and the queen larva and adult. Royal jelly is milky or light pale in color. Production. The queen cell is trimmed to the level of the royal jelly. After 2 or 3 days of grafting, larvae are gently removed with forceps and the royal jelly is removed with royal jelly spoon. This is stored in refrigerated conditions. In case of Apis mellifera, 200 milligrams of royal jelly is obtained from a queen cell. 412. 6. Pollen. It is collected by pollen trap from ingoing pollen foragers. Pollen is a rich protein source for human diet. 16.9 Role of honeybee in crop pollination. Qualities of honeybees which make them good pollinators. 1. Body covered with hairs and have structural adaptation for carrying nectar and pollen. 2. Bees not injurious to plants. 3. Adult and larva feed on nectar and pollen available in plenty. 4. Superior pollinators since store pollen and nectar for future use. 5. No diapause need pollen throughout year. 6. Body size and proboscis length suitable for many crops. 7. Pollinate wide variety of crops. 8. Forage in extreme conditions also, weather. Effect of bee pollination on crop. It increases yield, seed yield, fruit yield. In many crops it improves quality of fruits and seeds. Bee pollination increases oil content of seeds in sunflower. Bee pollination is a must in some self-incompatible crops for seed set. Scope of beekeeping for pollination in India. Total area under bee-dependent crops 50 million hectares at the rate of 3 colonies per hectare. 150 million colonies needed. In India only 1.2 million colonies exist. There is scope. Management of bees for pollination. Place hives very near the field source to save bees energy. Migrate colonies near field at 10% flowering. 413. Place colonies at 3 per hectare Italian bee, 5 per hectare Indian honey bee. The colonies should have 5 to 6 frame strength of bees, possess sealed brood, have young mated queen. Allow sufficient space for pollen and honey storage. 16.10 Summary. The science of beekeeping is known as apiculture. Apiculture makes use of low-cost, locally available resources that yield quick results. Honeybees are important for cross-pollination and crop yield improvement, which is a profitable venture with environmental, socioeconomic and cultural implications. However, due to some constraints such as improper harvesting, bee diseases, pests and predators, poisoning from agrochemicals and deforestation, beekeeping expansion has been slow. 16.11 Terminal questions and answers. 1. How long does a honeybee live? How long does a queen live? A worker bee typically lives 6 weeks during the summer, and during the time it has a variety of different jobs: nurse, undertaker, architect, cleaner, attendant, guard, forager. However, worker bees that live in the wintertime have a longer lifespan and can live up to 6 months. If they couldn't survive longer, they would all die, because the queen doesn't lay eggs in the winter. In the spring, when the queen begins to lay eggs, the overwintering workers transform back into job mode and start their typical 6-week lifespan. A queen bee lives anywhere from 1 to 5 years, and a drone lives about 6 months. However, a drone dies after he mates with another queen, so his lifespan could be much shorter. 2. How can you tell the difference between a worker bee, drone and queen bee? A worker bee is the typical bee that you would see, and the queen looks similar except her abdomen is about twice the length of a worker. This allows her to drop her abdomen into a honeycomb cell to lay an egg at the bottom. A drone bee is larger than a worker and is fuzzier. 3. How do bees carry pollen? 414. A honeybee carries pollen in specialized sacs on its hindlegs. You might have seen one with bright yellow packets of pollen on its legs as it returns to a hive from foraging. Many native, solitary bees are covered in stiff hairs to which pollen sticks. These bees can carry pollen all over their bodies, depositing it and helping with pollination as they move from flower to flower. 4. What are flowers that bees like? How can I help bees? It boils down to the fact that bees and other pollinators like native flowers, because that is what they are adapted to, that are bright, fragrant and have easily accessible and ample nectar. You can plant a pollinator garden in your yard or help support large pollinator plantings in the community. It is best to plant a variety of flowers, because different species are attracted to different species of flowers, and to plant flowers that bloom from early April through October. This allows for habitat and food throughout the growing season. 17.1 Objectives. Order: Hemiptera superfamily, Coccidae family, Laxiferidae genus, Laxifer species, Laca. Lac is an animal-derived natural resin. Lac insect secretes it. This technique is called lac culture and it involves cultivating insects in order to obtain lac. This involves the proper care of host plants, the regular pruning of host plants, the propagation of host plants, the collection of lac, and the processing of lac. To protect themselves, some hemipterans secrete lac over their bodies, such as Laxiferidae and Tachardinidae. The lac insect belongs to the superfamily Coccoidea of the order Hemiptera and is classified as a laxifer. 417. A total of 22 species have been recorded in the Indian subcontinent under the genus Laxifer. India is still considered to be the world's top lac producer. In the 16th century, Burma began trading lac. A lac culture dates back at least 4,000 years in China, and lac is used to dye silk and leather goods. Approximately 65% of the world's production is produced in India. India's total lake production is accounted for by Bihar and Jharkhand. 17.3 Lac insect and its life history. Hosts: plants such as Kusum, Schleicheratrihuga, Pallas bootifrondosa, Beer, Ziziphus jujube, Babool, Acacia arabica, Care, Acacia catshu, Arhar, Cahanusindicus. Laxifer equals Tachardia. Laca is commonly found on Ziziphus mauritiana, Bootimenosperma, Acacia arabica, Acatichu, Cahanuscahan, Ficus bengalensis, F. cunea, and F. religiosa. Biology of lac insect. 418. Adult lac insect. The lac insect, Laxifer laca tachardialaca, is commercially cultivated. Indian and Bangladeshi host plants include Beer, Ziziphus mauritiana, Pallas bootimenosperma, and Kusum, Schleichera oliva. Structure of male lac insect. It is larger and red in color. Typically, the body is divided into 3 parts: the head, thorax, and abdomen. Antennae and eyes are present on the head. Male adult insects lack mouth parts, so they cannot feed. There are 3 pairs of legs on the thorax. There may or may not be wings. Located at the posterior end of the body, the abdomen bears a pair of caudal setae and a sheath containing a penis. Structure of female lac insect. The size of the device is smaller. A pair of antennae and a single proboscis are located on the head. 419. There are no eyes. There are no wings or legs on the thorax. Female larvae lose their eyes, wings, and legs because they never move again after settling down, making these parts useless and eventually atrophying. There are 2 caudal setae on the abdomen. The bulk of lac is secreted by female lac insects. Life cycle. A female insect produces approximately 1,000 nymphs with black eyes and a deep red color. Larvae settle on a suitable plant host gregariously. Larvae begin secreting lac a day or 2 after settlement, except on the rostrum, spiracles, and abdomen tip. Thus, the insect is enclosed in a cell of lac that gradually increases in size as it grows. Before reaching maturity, the insect molts twice. Male larvae produce elongated lac cells, while females produce oval cells. Larvae lose their legs, antennae, and eyes after their first molt. A pseudo-pupal stage is reached after the third molt. The males emerge, copulate with the females, and then die. The female larvae never regain their appendages and remain under the lac cell until they become adults and reproduce. On the tree branch, lac secretion from adjacent cells coalesces to form a continuous incrustation as the lac insects remain close together. 420. Different forms of lac cell. 17.4 Host plant management. Production of lac crop is very much dependent on the species of host and obviously on the health status of the lac host. The health of lac host again depends on the fertility status of soil. Besides, there are a number of abiotic factors influencing lac production critically. Few of these factors are beneficial for lac production, few are beneficial for growth of the host tree, and rest is 421. Beneficial for both. Each of these factors are effective at a critical level for optimum production of lac. Thus, challenges of lac cultivation are different from that of general agriculture due to influence of so many factors. Management of soil and crop in a sustainable manner can only increase profitability in a lac production system. There could be a long list of such factors. These are crop rotation, irrigation and drainage, plant breeding, plant physiology, soil classification, soil fertility, weed control, insect and pest control. Therefore, maximization of production is possible when all these factors are manipulated in a compatible way to harness the positive interaction of factors on lac yield. Therefore, maximization of production is possible when all these factors are manipulated in a compatible way to harness the positive interaction of factors on lac yield. Canopy management. In the natural stand of beer, plants with spreading, semi-erect, erect and umbrella bushy type shaped are observed. Spreading type trees are usually vigorous in nature and good producers of lac, provided the insect settlement is not affected due to poor light interception. Erect type trees are most consistent yielders due to good distribution of sunlight and ventilation throughout the canopy. Umbrella shaped or bushy trees are poor lac yielders due to poor distribution of light and improper ventilation inside the canopy.